Hello everyone, and welcome to Lecture 5 of Unit 1. In this chapter, we are going to start getting into the details about prokaryotic cells. We're going to talk about how they're structured, we're going to talk about different functions of those structures and how these organisms function as a whole, as well as start to get into a little bit about how these organisms can affect us. You've got your lesson objectives here. I recommend checking them out before you start this lecture so you know specifically what you're listening for, as well as checking them out again to make sure that you can meet them after listening to this lecture. To start us out and kind of set the stage for the rest of the lecture, I want to first talk about the 3 different domains of life that we can find on this planet. So domain is the highest level of classification for living organisms. It's above kingdom, and it's based on the type of cell and genetic sequences. When we classify life on Earth, we talk about domains, kingdoms, phylums, classes, orders, families, genus, and species. So when we're talking about domain, we're talking about this level right here, the highest level of classification of living things on Earth. And there are, like I said, there are 3 domains of life on Earth. One of these domains is called eukarya. Eukaryotes are things like people, plants, animals, algae, slime molds, fungi, etc. We study some eukaryotes in microbiology. Not a lot of them, but a few of them. And to me, eukaryotes are not the most exciting thing in the world. To me, I think they're kind of meh. However, there are 2 prokaryotic domains, the bacteria and the archaea, and personally, I find these a lot more exciting than eukaryotes. Bacteria have a lot of significance medically and ecologically. They have an incredible amount of diversity, and like we've talked about previously, they really are part of the basis of life on Earth. So we've talked about how microbes make up the basis of all life on Earth, and some of the most significant microbes are the bacteria. Archaea are another group of microbes. They are also prokaryotes, so they are single-celled. They have no membrane-enclosed organelles. Same with bacteria, single-celled, no membrane-enclosed organelles. They oftentimes live in very extreme environments, not always, but oftentimes they live in places that are very hot or very dry or very salty, very acidic, very cold, etc. They oftentimes live at the extremes of what we would consider livable or survivable environments on planet Earth. Archaea are very interesting in the fact that they share characteristics of both bacteria and eukaryotes. So they are like bacteria in the sense that they are prokaryotic, they are single-celled, they have no membrane-bound organelles, but in terms of certain genetic sequences, certain parts of their DNA, certain parts of their RNA, they're a lot more similar to the organisms we find in the domain eukarya. So they're a lot more similar to eukaryotes in some of their genetic sequencing. So they are very intriguing in that they are kind of a bridge organism between bacteria and eukarya. And what I mean when I say that is that there was once a common ancestor to all living organisms on the planet. At one point in time, a very long time ago, there was one single organism that would go on to have descendants that would eventually develop into all of the life forms on the planet. And the organisms today that probably resemble that common ancestor the most are likely found in the group Archaea. So Archaea are this group of organisms that have, like I said, characteristics of both bacteria and both eukarya. And so they are very similar to, or at least probably the most closely related that we can find in this day and age to that one common ancestor that ended up giving rise eventually, we're talking billions of years, to all different life forms on the planet. So just to recap here, there are 3 different domains of life on Earth. We find microbes in all of the domains. We can find them in bacteria, archaea, and eukarya. We are going to spend most of our time in this class talking about the bacteria. Both of the bacteria and the archaea domains contain prokaryotic organisms. And then the domain eukarya, of course, contains eukaryotic organisms. Prokaryotes are always single-celled. They do not have membrane-enclosed organelles. And like I've said, they form kind of the basis of life on Earth. Eukaryotes can be single-celled or multi-celled. They are also, of course, important to life on Earth, but we're going to spend most of our time in this class focused on prokaryotes, especially the bacteria. Now that we've talked about the 3 domains of life on Earth and gone over a little bit about each of them in terms of their characteristics, what their cells are like, etc., we are going to spend the rest of the lecture talking about the characteristics of prokaryotic cells in detail. So we will talk about eukaryotes as well. That will be in another lecture in another chapter. But for this lecture and this chapter, we are just focusing on the characteristics of prokaryotic cells. I've mentioned this a couple of times previously, but just again to recap, to hit on this because it's a very important point. There are 3 main characteristics of prokaryotic cells. They are always single-celled, they do not have a nucleus, and they do not have any other membrane-bound organelles. So at the top here, you're looking at a prokaryote. At the bottom, you're looking at a eukaryote. You've probably spent a lot of time actually looking at eukaryotic cells and studying them previously. General biology, anatomy and physiology, all of those classes are focused on eukaryotic cells. But this class is focused on prokaryotic cells, and we are going to spend the rest of this lecture and really the rest of this class talking about these types of organisms and how they affect us. Oftentimes, when people talk about prokaryotes, they say that these are simple cells, they're just kind of basic cells, they don't have a lot of parts to them, and they kind of brush them off a little bit. I'm going to show you that this is not the case, and that prokaryotes are quite complex, actually, have a lot of different important physical features that can affect humans quite a bit. And so we are going to go through the different parts of prokaryotic cells, starting from the outside and working our way in. There are different ways to do this. We could start from the inside and work our way out, but I tend to find it goes a little bit better if we start out looking at the external structures and then working our way inside. So the next several slides, we are just talking about things that we can find on the outside, and I will let you know when we switch to talking about things that we find on the inside. The first external structure that we will talk about is called the glycocalyx. So the glycocalyx is the outermost covering of a prokaryote. In this diagram, it is pictured in light pink. And there are 2 general types of glycocalyces that prokaryotes can have. Occasionally, we will find a prokaryote that has both, but for the most part, prokaryotes will have either a slime layer or a capsule. So these are both types of glycocalyces, but there are just 2 different varieties. A slime layer is kind of exactly what it sounds like. It's a loosely arranged, not very structured kind of slime that covers the bacteria. Usually it's pretty sticky. A lot of times it has a lot of polysaccharides in it, and as you guys have probably experienced in life at this point, sugar tends to be sticky. So lots of times the glycocalyx is a little bit sticky, and the slime layer is a form of glycocalyx, so it's a little bit sticky. And basically this type of glycocalyx just kind of seeps out from the organism and surrounds it in this coat of slime. The other type of glycocalyx is a capsule. A capsule is a very tightly packed structure. So it's tightly packed The molecules that make it up are oftentimes regularly arranged in a specific pattern. And it has a definite structure and definite shape to it. It also is made primarily of polysaccharides. It also may have some proteins thrown in it. So both slime layers and capsules mostly are made of polysaccharides, may have some proteins in them. And the function of the glycocalyx, whether it is a slime layer or a capsule, is of attachment. So it will help the prokaryote stick to things in its environment. Protection, especially from the immune response, if it happens to be living in our body or in another animal's body. And this outermost covering, this glycocalyx, a slime layer or a capsule, will help protect the prokaryote from our immune response to it. It can also help prevent the bacteria or the prokaryote from drying out. It can help prevent a loss of nutrients. So the slime layer and the capsule can do all three of those things, although the capsule is a little bit better at doing protection from drying out and from loss of nutrients. So what I wrote there was just the capsule is better at these two things, but the slime layer can do them too. And one final thing about the glycocalyx is that it oftentimes helps the prokaryote become more pathogenic. So it aids in pathogenicity, which basically means it makes the organism more capable of causing disease. If a bacterium has a really good slime layer or a really good capsule, sometimes it can be very, very difficult, again, for our immune response to deal with this type of organism. And as a result, the organism is able to cause disease or cause worse disease than it normally would be able to. Now that we've talked a little bit about the structure of the glycocalyx and some of the general things that the glycocalyx can do for a prokaryote, I want to give you a real-world example about why this structure is so important and what it can actually do to affect the environment and humans. One of the things a glycocalyx is often involved in, not always, but often, is in forming what we call a biofilm. We will talk about biofilms much more extensively in upcoming units, but for now, we are going to define a biofilm as many microbes growing together on a surface in a matrix. And a matrix is basically a bunch of glycocalyces all joined together. Sorry, my pen was being difficult there. So a matrix, like I said, is basically just a bunch or a combination of glycocalyxes from individual prokaryotes that have all joined together in a big structure. And that structure is now just protecting those individual organisms and allowing them to essentially work as a group instead of as individuals. We can find biofilms in various places in the world. One place you may have actually seen them before, even if you didn't realize it, is somewhere out in the environment. They are oftentimes found on various rocks in lakes, in rivers. We can sometimes find them on stream bottoms. We might even find them out in the ocean in certain circumstances. But basically, if you look at these top two pictures and you see this kind of green, slimy stuff growing on these rocks, and this is kind of a zoomed in version of what is growing on there, all of that green stuff, if you were to pick it up and feel it, it would probably be very slippery, maybe even a little bit sticky. And the reason for that is because essentially what we have here is a bunch of glycocalyxes all joined together, attaching themselves to that rock. And within that large mass of glycocalyx, there are a bunch of individual organisms. And that is what we call a biofilm. So like I said, you guys have probably actually seen these before, or you maybe even felt them before out in the environment. You just didn't necessarily know that that's what you were feeling or touching at that point in time. We can also find biofilms in the human body. Biofilms can really develop anywhere in the human body that there is some kind of surface to attach to, but there are a couple of common places or places that they're more likely to form. All of us have a biofilm on our teeth, and the main purpose of brushing and flossing our teeth is to kind of disrupt or destroy that biofilm, at least to the best of our ability, so that it doesn't start to take over and cause damage to our teeth, to our gums, and potentially to our cardiovascular system. So basically, when you see someone and they have a lot of tartar or a lot of plaque that has built up on their teeth, that is really a result of the overgrowth of a biofilm that's in their mouth. Like I said, all of us have it. We can't ever really get rid of it. We just have to control it the best we can. And if we don't control it, then it gets out of control and it starts to kind of build up on our teeth and cause damage in our mouth. Oftentimes, implanted medical devices will also develop biofilms. Again, this is not like always, it doesn't always happen, but a lot of times, or at least a significant number of times, when something gets implanted in the body, it can make someone more prone to developing a biofilm on that implanted device. Whether it is there permanently, like a screw or a pin, or something that's designed to maybe hold a bone together or help stabilize a joint, or even something more temporary like a catheter. Anything that we implant in the body provides another surface for microbes to grow on. And so it is not uncommon for people who have things implanted in their body to develop a biofilm on that implanted device. Some people get biofilms that develop in the urogenital tract. When someone gets repeated urinary tract infections, it is often because they have developed a biofilm in their urogenital tract. That's not always the case. Not all UTIs are caused by biofilms or are the result of biofilms. But if someone is getting repeated urinary tract infections that antibiotics just don't seem to quite ever cure or get rid of the infection for them, it may be because they've developed a biofilm in that region of their body. Some people with certain lung conditions are also more prone to developing biofilms in the lungs. It could happen to any of us, but certain conditions make people more prone to developing biofilms in the lungs. In terms of the significance of biofilms, especially when we're talking about in the human body, they are particularly problematic for two main reasons. One, they may be resistant to different types of medications or drugs that normally work on the microbes that are making up that biofilm. And two, they are very, very difficult to destroy. Once these prokaryotes join together, basically they join forces and they all are covered in this matrix, they're protected by that matrix essentially, then they oftentimes become a lot more drug resistant and they're very, very difficult to actually fully get rid of, even through physical methods. A great example of that is our teeth, right? We all have to brush our teeth, hopefully at least 2 times a day. And don't forget to floss, that's important too. But we all have to control that biofilm in our mouth multiple times a day even. Obviously, it's something we have to take care of our whole life once we develop teeth and hopefully keep our teeth for the rest of our life. But we have to manage that biofilm. We have to control that biofilm because we can't really ever get rid of it. If we could get rid of it, we wouldn't necessarily have to brush our teeth as often. But we can't get rid of it. That biofilm is always there. Even if we brush and floss on a regular basis, the bottom layer of that film basically never really goes away. And so we have to continuously take care of that and manage that and kind of knock the levels of biofilm down to make sure that it doesn't overgrow and doesn't cause problems. So we will talk about, like I said, biofilms in more detail in subsequent units, but for now, that covers our discussion of the glycocalyx and the significance of just this outermost layer of the prokaryote. The next external structure that we will talk about is the cell envelope. The cell envelope is found just underneath the glycocalyx, whether that is a slime layer or a capsule, and it is composed of two structures. It is composed of a cell wall and a cell membrane. So remember this, it goes from the outside to inside, goes glycocalyx and then cell envelope. And the cell envelope is made of two structures, the cell wall and the cell membrane. The cell wall is a rigid structure. Its primary function is to provide shape to the prokaryote as well as support, and it is composed of a molecule called peptidoglycan. So you're looking at that molecule right here. Peptidoglycan is basically a sugar protein. That's why it's called the peptido and the glycan. The peptido part is the protein part and the glycan is the sugar part. And this peptidoglycan has multiple subunits that essentially just join together. And it's kind of this repeating subunit pattern that just all joins together and forms a net or a mesh-like structure around the bacterium and helps provide, like I said, shape and support to the organism. Underneath that cell wall, so underneath that layer of peptidoglycan, is what we call a cell membrane. The cell membrane is a flexible structure. Its primary function is to determine what comes in a cell and what goes out of a cell. It is composed of phospholipids, proteins, carbohydrates, etc. You have studied the cell membrane probably quite a bit in previous biology classes, or at least in your introductory biology class. So this is just a typical phospholipid bilayer, same basic structure that you've learned about before. This slide is essentially just a close-up view of the cell envelope. Right here, you have the rigid cell wall. This is the peptidoglycan, just the layers of peptidoglycan kind of joined to each other, forming this mesh or this net around the bacteria. And then underneath that, you have the cell membrane, which is more flexible. It has different types of proteins and carbohydrates in it. Sometimes it will have these kind of channel-like proteins in it that help facilitate movement in or movement out of the cell for different types of molecules or chemicals. But this one, I just wanted to give you this as another view, another way to look at the cell envelope. Within the domain bacteria, there are two main types of cell envelope arrangements. There are cells that have what we call the gram-positive arrangement, and other cells that have what we call the gram-negative arrangement. In the gram-positive cell envelope, the structure goes like this. There is a cell wall composed of a very thick layer of peptidoglycan. That peptidoglycan layer also has teichoic acid and lipoteichoic acid interspersed within it. Underneath that very thick layer of peptidoglycan is the cell membrane. This type of cell envelope arrangement stains purple when we perform what's called a gram stain. I'm not going to go into the gram stain in this lecture because you guys are going to learn about it extensively in lab. But for now, what you want to focus on is the structure of the gram-positive and the fact that it will stain purple. So gram-positive has a very thick cell wall, a thick peptidoglycan layer with teichoic acid and lipoteichoic acids interspersed within it. And underneath that very thick cell wall or peptidoglycan layer is a cell membrane. The other main group of bacteria is what we call the gram-negative bacteria. The gram-negative cell envelope is a little bit more complex. It starts out with an outer membrane. This outer membrane has porins, which are protein channels that allow certain things in and maybe help certain things pass out. It also has lipopolysaccharide, or LPS. Lipopolysaccharide is a very important structure, at least to humans, because it can provoke a very strong immune response. It's important to the bacteria because it makes up most, if not all, of the outer membrane for these gram-negative organisms, these types of bacteria. But it's significant to us not just because of the fact that it makes up the outer membrane, but because it can provoke a very large immune response. Underneath the outer membrane is the cell wall. The cell wall in gram-negative organisms is much thinner than the cell wall in gram-positive. So it's still composed of peptidoglycan, but it's just a thin layer of peptidoglycan instead of a thick layer. And then underneath the cell wall is the cell membrane. This type of organism, when we perform the gram stain, stains pink. Again, you will learn about the details of the gram stain in lab, so I'm not going to go over that in lecture as well. But for now, this is the information that you'll want to focus on. You want to focus on the structure or the composition of the cell envelope and the fact that the gram negatives stain pink. What I've got for you here is a detailed visual of what we were just talking about on the previous slide. On the left-hand side, we have the structure of the gram-positive cell envelope. On the right-hand side, we have the structure of the gram-negative cell envelope. So remember, all of this is underneath the glycocalyx in both of them. And so essentially, for the gram-positive, what we would have if we were going outside to inside is we would have the glycocalyx, then we would have a very thick layer of peptidoglycan, so that's the cell wall. Underneath the peptidoglycan, we would have a small periplasmic space, which is just the space in between the cell wall and the cell membrane. And underneath that, we would have the plasma membrane or the phospholipid bilayer, also called the cell membrane. Sorry, my dog is leaving the room. And like I said, within the plasma membrane or the cell membrane, you can also find certain proteins. Those proteins serve to help allow things come into the cell or go out of the cell. They help provide structure to the cell membrane. They help serve as certain recognition sites, etc. The gram-negative cell envelope starts out with the outer membrane, which is right here. Remember, within the outer membrane, you have porins, which are these protein channels that determine what comes into the cell, and to a certain extent, what might go out of the cell. Underneath that, you will have a very thin layer of peptidoglycan. Underneath that, you will have the cell membrane. So for the gram-negative cell, you would have a glycocalyx up here, the outer membrane here with its porins and its lipopolysaccharide, the thin layer of peptidoglycan, and then the cell membrane. We have just spent the last 15 to 20 minutes talking about just the outermost layers of prokaryotes, and hopefully you're starting to see why I think these are such exciting organisms and why I don't agree with people when they say that prokaryotes are simple cells and they're not as complex as eukaryotes. I personally think that they are as complex and just as significant as eukaryotes. And I also promised I would tell you when we were moving from the outside of the cell to the inside of the cell. So on these next several slides, we are going to start talking about the internal components of the prokaryote. So we're switching from external to internal at this point in the lecture. One of the major internal components of the prokaryotic cell, at least in terms of volume, is the cytoplasm. The cytoplasm is kind of a jelly-like substance that fills up the internal compartment of a cell. It is made primarily of water, and it also has various macromolecules and other cell structures. So within the cytoplasm, you will find a bunch of polypeptides, which are the early parts of proteins. You'll find polysaccharides, you will find nucleic acids, you will find lipids, you'll find all kinds of the different macromolecules in the cytoplasm. And of course, you'll find the other internal cellular structures of the organism. In terms of the function of the cytoplasm, in eukaryotes, most of their metabolic reactions take place within their specific organelles. They have things like the endoplasmic reticulum, or they have the Golgi apparatus, they have the nucleus, etc. But remember, prokaryotes do not have a nucleus, and they don't have any other membrane-bound organelles, which means that almost all of their metabolic reactions are going to have to happen in the cytoplasm. So the cytoplasm is a very busy and very significant structure for prokaryotes because all of the metabolic reactions that they need to stay alive occur there. The cytoplasm typically holds the raw materials that the organism is going to use to carry out these metabolic reactions. So it's going to hold the sugars that it will break down to create ATP. It's going to hold the amino acids that the organism is going to use to make proteins. And so essentially all of the metabolic reactions and all of the raw materials that the organism needs for those reactions is in the cytoplasm, which makes the cytoplasm very significant for prokaryotes. Within this large internal component of the prokaryotic cell, we will also find the cytoskeleton. Now, the cytoskeleton is made of various proteins. In prokaryotes, it is made of these three main proteins called MREB, FITZ or FTSZ, and CRES. In eukaryotes, there is also a cytoskeleton, but the proteins are different. We'll talk about the exact composition of the eukaryotic cytoskeleton in the next lecture, but for now what you want to focus on are these three proteins. Sometimes other cells, or sometimes certain cells have other proteins as well, but these are the three main ones. In terms of the function of the cytoskeleton, it is involved in providing shape and structure. So it works basically in conjunction with the cell wall to provide shape and structure to the cell. And it especially is important in providing structure to internal cellular components. It is involved in metabolic reactions and peptidoglycan positioning. So while most metabolic reactions happen in the cytoplasm, the cytoskeleton is there within the cytoplasm and can help these metabolic reactions take place. And part of those metabolic reactions involve positioning the peptidoglycan that will make up the cell wall that will give shape and support and protection to the cell overall. And the final main function of the cytoskeleton is with cell division. Prokaryotes divide through a process called binary fission. We'll talk about that in, again, subsequent units and subsequent lectures. But for now, what you're focusing on is just the fact that the cytoskeleton will help this process called binary fission take place. Within the cytoplasm, one of the biggest structures that we will find is the chromosome. So bacteria, prokaryotes, just like eukaryotes, have genetic material that they use to create proteins with and that they use to pass information on to the next generation of cells. And prokaryotes have typically one chromosome. It is circular most of the time. And the chromosome contains the necessary genetic information for the organism to survive. So all of the information that the organism needs to survive, to do things like go through glycolysis, to create peptidoglycan, to make its cell membrane, to do all of its other metabolic reactions, etc. All of that information is contained within the genetic material of the chromosome. Prokaryotes do not have a nucleus, but they often have what's called a nucleoid region. And in the nucleoid region, that is where we typically find this one singular chromosome that prokaryotes have. Another kind of unique thing about prokaryotes is that they oftentimes, not always, but oftentimes have what we call extrachromosomal genetic information. Extrachromosomal just means outside of the chromosome. And that extra chromosomal material is often in the form of what we call a plasmid. Plasmids oftentimes contain optional or nice to have genes that aren't necessary for survival, but give the organism some kind of advantage. So as an example for that, let's say there is a type of prokaryote or a type of bacteria that normally uses glucose as the source of energy necessary to create ATP. And that is fine. This organism goes through its metabolism, it breaks down glucose, it creates ATP. And the genes that are necessary to carry out all of those metabolic reactions are contained on this chromosome. But let's say this individual acquires a plasmid that contains the genes that would allow it to break down lactose as well. And so this organism is put into an environment where both glucose and lactose are available. Now, because this organism has this plasmid with the genes that will let it break down lactose, it can also use lactose as a source of energy to make ATP. So basically, the chromosome contains the genes that are needed to break down glucose, and that is fine. That will allow the organism to survive and do just fine. But this particular plasmid contains the information that allows this individual bacteria to also break down lactose and use that as a source of energy to make ATP as well. That means that this individual has an advantage because now it has two potential sources of energy rather than just one. So what I've given you here is an example. Not all plasmids will contain genes to break down lactose, etc. That was just an example. And what I was using it to do is just illustrate the fact that plasmids oftentimes contain genes that are useful to the organism, that give them an advantage in a certain environment, but aren't necessary to stay alive. In this example, if you took the plasmid away, this organism would still be fine because the chromosome contains the genes that are necessary for survival. But the plasmid just gives it an extra advantage in a certain environment. Most of the time, plasmids are circular in nature, and bacteria or prokaryotes can have single or multiple plasmids, or they can have none at all. Some bacteria have no plasmids. And another interesting thing about plasmids is that sometimes a bacteria will gain or lose plasmid throughout its lifetime. So let's say a bacteria starts out with no plasmid, but it gains a plasmid because it's lucky, and that plasmid gives it an advantage. But then something goes wrong and it loses its plasmid, and it just goes back to being a normal bacteria without a plasmid. So we've had a lot of information on this slide. Some of it, especially the information about plasmids, may have been very new to you. So I'm just going to recap here. All bacteria have one chromosome. That chromosome is usually circular, and oftentimes it's found in the nucleoid region. That chromosome contains all of the genetic information that the bacteria absolutely must have to stay alive. So it contains the necessary information. Some bacteria may also contain one or more plasmids. Plasmids are extrachromosomal, meaning they are found outside of the chromosome. Usually they're circular in nature. And oftentimes these plasmids give the bacteria some type of advantage in its environment. The example I used was that maybe a plasmid would allow the bacteria to break down lactose, which means that it has an extra source of food aside from just glucose. But really, you could be talking about any type of advantage. Maybe it allows the bacteria to produce a type of pigment that makes the bacteria able to gather sunlight and get energy from that source. Maybe a plasmid has genes on it that allow the bacteria to reproduce faster. So it allows the bacteria to make copies of itself faster than other bacteria and gives it that advantage in that regard. Basically, the plasmid could have any number of different genes on it, and those genes are advantageous to the bacteria, but not required for survival. They give it a boost, but they aren't absolutely needed for the bacteria to survive. Another important structure that we will find within the cytoplasm of the prokaryote is the ribosome. Ribosomes are small structures that are made of RNA and protein. They have two subunits, what we call a 50S subunit and a 30S subunit. The 50S is the big one, so I'm going to put that one up top, 50S, and then the 30S is the small one. And in everywhere except microbiology, 50S and 30S would add up to 80S, but in microbiology, that actually adds up, in this circumstance, to 70S. So that's not a typo, that's not a mistake. The total size of the prokaryotic ribosome is what we call 70S. So you will want to know these numbers. So the 50S and the 30S are important, but the 70S is the most important. You'll want to focus on that number for the most part. The function of a ribosome is to basically provide space for protein synthesis. So in between the big subunit and the small subunit, there is a space, and messenger RNA is able to fit in this space. Transfer RNA is able to come over and attach to the messenger RNA and bring the amino acids that are necessary to make proteins and allow them to be strung together. So it works the same basic way that it does in eukaryotes, if you think back to general biology. And don't worry, we will spend a significant portion of time in our next unit talking about the details of this. But for now, what you want to focus on is prokaryotic ribosomes are 70S in terms of their size, and their function is protein synthesis. They are the site that allows protein synthesis to happen. Now, other things we might find in the prokaryote are called inclusion bodies. So all prokaryotes are going to have cytoplasm, they're going to have a cytoskeleton, they're going to have genetic material, they're going to have ribosomes, and then some may also include inclusion bodies. Inclusion bodies are structures that are found in the cytoplasm that are often used for nutrient storage and or movement. Typically, they are not membrane bound. There are a few exceptions to that. So occasionally we will have a structure, an inclusion body that does have a membrane that surrounds it. But most of the time, inclusion bodies that are found within the cytoplasm of prokaryotes do not have membranes surrounding them. There are some examples that are kind of fun to talk about. One are magnetosomes. Magnetosomes are a specific type of inclusion body that are filled primarily with iron, and they allow the bacterium to orient itself on a north-south axis. So they essentially act like little compasses within the cytoplasm of bacteria that tell them where north is, where south is, and then also where east and west would be. Some bacteria have what we call gas vesicles. Gas vesicles are used to move up and down in liquid. So this is especially important in bacteria that live out in the environment in something like a lake or a stream or a pond or something like that. Basically what happens with these is when the bacteria needs to move up towards the surface, it creates a gas vesicle that gets filled usually with nitrogen gas, and that allows the bacteria to float up towards the surface or to the surface of the body of water. And then if the bacteria needs to move down closer to the bottom of that body of water, it will cause that gas vesicle to deflate. It will release that nitrogen gas, and then it will start to sink. It will start to go to the bottom of that body of water. And then many bacteria have inclusion bodies that serve as sponge storage. We talked about the importance of the sponge elements. Remember, sponge elements stands for sulfur, phosphorus, carbon, nitrogen, and hydrogen, and oxygen as well. I forgot that one. And many organisms will store certain of these elements, especially sulfur, phosphorus, carbon, and nitrogen, within their cytoplasms to use them later, just in case conditions get a little bit rough and they are lacking in certain nutrients. So there are other types of inclusion bodies, of course. I've just included some of the most common and some of the most interesting ones in the slide to help illustrate about the importance of inclusion bodies and what they can do for the organisms. So we have talked about the external parts of the prokaryotic cell. We've talked about the internal parts, and now we are going to switch gears a little bit and talk about special structures, especially things that oftentimes will stick out of the prokaryotic cell. The next several slides will go over things like the flagellum, the pilus or the pili, and some other important structures as well, so that you understand these appendages that often will stick out from the surface of the cell. Our first appendage that we'll be talking about is the flagella. Flagella are long, thin protein filaments that basically stick out from usually one end of the bacteria. Not all bacteria have these. Some of them do, many of them do not. And what the flagella does for those bacteria that do have them is allow the organism to propel itself forward or backward. So it doesn't really allow for lateral movements per se, it allows for forward and backward motions. And it kind of depends on which way the flagellum is turning. If the flagellum is turning clockwise, the bacteria moves forward. If it is moving counterclockwise, I was about to say anti-clockwise, which I guess would be correct too, but if it's moving counterclockwise, then the bacteria moves backwards. Now, bacteria that have flagella can have either one flagella or multiple flagella, and the different arrangements they have are illustrated here on the right-hand side. If a bacterium has just one flagellum, that is called monotrichous. If it has a clump of flagellum on one end, that is called lophotrichous. If it has two flagellum on kind of either end, that is called amphitrichous. And if it has multiple flagella kind of all over the surface, almost it looks like hair coming out of the bacterium, that is called a peritrichous flagellum. Flagella are important, one, because it allows these bacteria to move, and that is significant both environmentally for allowing them to move through bodies of water or other types of liquids, but also in our bodies as well. If a bacterium has a flagella, it makes it easier for that bacteria to move throughout our body, especially in places where there's liquid, which our body has a lot of. So flagella are significant medically because it allows a bacteria to move throughout our body a little bit easier. And looking at the arrangement and the number of flagella can also tell us at least a little bit about what type of organism might be infecting us. So, for example, in our intestines, there are a lot of gram-negative rod-shaped organisms, and some of them are pathogenic, some of them are not. And if we were to try to identify the different types or different species of these gram-negative rod-shaped organisms that we find in our intestines, one of the things that we would look at is if they have flagella, and if they do, how many and what arrangement they're in. Because the different species that live in our intestines have different numbers and different arrangements of flagella, and that will tell us what types of bacteria or what types of prokaryotes we are dealing with in certain circumstances. Our next important structure that we'll talk about are the fimbre. Again, not all bacteria have these, but many of them do. And fimbre are kind of medium-length proteins that stick out of bacteria and are used for adherence. They allow the bacteria to stick to things in their environment. I like to say they kind of act like Velcro. What they do is help keep the bacteria attached or anchored to a host or a specific location in the environment. And these structures are particularly useful in places where there is a lot of flowing liquid or liquid that is moving through a particular environment. One example, this example here, is an organism called Neisseria gonorrhea. It causes the sexually transmitted infection we call gonorrhea. And this particular organism has some really, really sticky fimbre that allow it to persist in the urogenital tract of humans in ways that other bacteria can't necessarily. So one of the characteristics of the urogenital tract is that there are a lot of times when it is flushed out fairly regularly, right? So during the day and sometimes during the night, people urinate, and that serves in part with getting rid of waste products in our body, of course, but in part it helps flush out this particular area of our body. And many bacteria that are in that region of the body will be washed out as a result. They will get washed out of the body, and therefore they won't have a chance to infect. However, this organism in specific, and this is just an example, again, there are plenty of bacteria that have fimbre that I'm not talking about right now, but it's just that this example happened to be very good. This bacterium that causes the sexually transmitted infection we call gonorrhea happens to have some particularly sticky fimbre that allows it to stick in the urogenital tract and not be washed out as easily as other bacteria. Some will, of course, be washed out, but many will still be able to stick in there and persist and continue to infect the body because of these fimbre. Another structure that bacteria may have is something called a pilus, or the plural of that is pili. And these are protein tubes. They can be long, but they're oftentimes relatively short. And they can either be used for movement or for sharing genetic material. So there are multiple different types of pili, and certain types are used for movement. In this case, unlike the flagella that kind of propels the bacteria through the environment, these pili are used more in kind of a walking or crawling motion. So it's not truly walking and not truly crawling, but it's a very similar motion to as if the organism were crawling through its environment. So when an organism moves, that is called taxis. So I'm just going to write that over here, movement. The technical word for that, or the scientific word for that, is called taxis. And organisms tend to move in response to chemicals or light. So if an organism is crawling through its environment in response to chemicals, that would be called chemotaxis. And if an organism were crawling through its environment in response to light, that would be called phototaxis. These terms are true for movement caused by flagella as well. I just happen to be talking about them on this particular slide. So pili, certain types of pili, can be useful for taxis, which is movement, and these organisms tend to crawl through their environment in response to either chemicals or light, the same way an organism might use its flagella to propel itself through the environment in response to chemicals or light. Now, another type of pilus will allow organisms to share genetic material with each other. So these types of pili are oftentimes very long, and they stretch from one organism to another. And they're kind of this hollow tube that is used to pass information from one organism to another. Most of the time, the information that is passed is a plasmid. And we talked about plasmids on a previous slide. But basically what you're looking at here is you have bacteria A on the right and bacteria B on the left. Bacteria A has a chromosome and a plasmid. Bacteria B just has a chromosome. So what bacteria A is going to do is it's going to create this pilus, and this long, hollow tube of protein, and it's going to reach over to bacterium B. What this pilus basically does is allow the cytoplasms of the cells to be connected. Now this cell, bacteria A, is going to send over part of the plasmid to bacteria B so that bacteria B can get a copy of that plasmid and have the same traits or the same characteristics that bacteria A does on that plasmid. So I don't have purple on this pen, so I have to draw it in green. But basically what's happened here, this process, part of the plasmid from bacteria A has been sent over through this pilus to bacteria B. The rest of it has been replicated. And so now each of them have a full copy of the plasmid, and bacterium B now has all of the same information that bacterium A has. The process itself of sharing information this way is called conjugation. And this is another thing that we will talk about in depth in a subsequent unit or in a future chapter. And for now, what you want to focus on is just that conjugation is one of the things that a pilus can be used for. And what happens in conjugation is that genetic information from bacteria A is shared with genetic information to bacteria B. That's the big thing to focus on at this point in time. All of the little details about how this happens will be talked about in another unit. The last structure we will talk about isn't really an appendage, but it is something that certain bacteria can make under certain environmental conditions. This structure is called an endospore, and endospores are protective structures. They are not reproductive structures, so that's something to really keep in mind. Endospores are protective structures, not reproductive structures. And this structure is developed in response to harsh environmental conditions. So not all bacteria can make endospores, but for bacteria that can make endospores, if they realize they are in a harsh environmental condition, for example, it is too salty, it is too hot, it's too cold, there's radiation, there's not enough nutrients, basically anything where they realize they're not going to survive it, what they're going to do is they're going to make a copy of their chromosome, and they're going to take that copy, usually, and shove it off to one side of the cell. They're going to then surround that copy of the chromosome with several different layers of chemicals that create what is almost an impenetrable wall around this bacterium. It's like a fortress around this chromosome. Now, because environmental conditions are bad, the rest of this cell is going to fall apart. Basically, that cell is going to fall apart, it's going to disintegrate, and what will be left is just this little tiny, usually round structure that is protecting the chromosome. So all that's in there is the chromosome, a little bit of cytoplasm, some nutrients, and some ribosomes. And these structures, these endospores that have been created to protect the chromosome, are extremely durable. They are resistant to most environmental conditions, so they're resistant to most types of heat, they're resistant to radiation, they're resistant to chemicals to a certain degree, they're resistant to cold, they're resistant to salt, and they can survive 250 or more million years in the environment and still potentially turn into a bacterium when conditions get better. So this is another slide where there's a lot of kind of new information and new vocabulary terms, and I just want to recap here. Some bacteria, not all, but some bacteria, are able to produce a structure called an endospore. Endospores are used to protect the chromosome in harsh conditions. If a bacterium realizes that it's in a harsh condition, that it's not going to survive, it's going to copy its chromosome, and it's going to shove that copy of the chromosome off to kind of one side of itself. It's going to surround that copy of the chromosome with many different layers of molecules or different types of chemicals that will create a very, very protective shell around that chromosome. That bacteria is going to die. It's going to degrade and fall apart. And what will be left is what we call the endospore. So this is the endospore. And the endospore is a very tough, durable structure that can survive in the environment, at least in many cases, 250 million, potentially more years. And still, if environmental conditions get better, turn back into what we call a vegetative bacterium. So a vegetative bacterium is just a normally metabolically active living bacterium. So endospores will protect the chromosome for potentially 250 million years, maybe even more than that. And then if conditions improve, if they get put into an environment where they have enough nutrients, it's not too hot, it's not too cold, it's not too salty, it's not too filled with anything else, it's just a good environment for them, they can germinate and turn into what we call a vegetative bacterium, which would just be a normal metabolically active bacteria. The last thing we'll talk about in this lecture are some of the basics regarding prokaryotic cell shapes and arrangements. Prokaryotes come in three basic shapes. They are either round, which is also called coccyx or cocci. They are rod-shaped, which is called bacillus or bacilli, or they are spiral-shaped, and there are two different types of spirals, spirillum or spirochetes. So looking at our picture here, our round bacteria are called cocci. Our rod-shaped bacteria are called bacilli. And then our spiral-shaped bacteria, the main ones that we can find, are the spirillum and the spirochetes. There are some other shapes as you're looking at here. We do have a couple of other special shapes that we can find, but the majority of bacteria that you're going to encounter, especially in the healthcare professions, are the cocci, the bacilli, the spirillum, and the spirochetes. Now, for the cocci and the bacilli, they can come in different arrangements. Spiral-shaped bacteria are always what we call single. So a spirillum will always just be one, and a spirochete will always be one. But cocci and bacilli can come in different types of arrangements. Any of these can be singles, just individual bacteria out there. But cocci and bacilli can also form diplo arrangements. Diplo means two. So if you look at our picture of our cocci, we have the diplococci, which means two joined together. We can also have diplobacilli, which are two rod-shaped organisms joined end on end. Both bacillus and cocci can also form a strepto arrangement. So streptococci is a bunch of cocci all joined together in a chain. Streptobacillus or streptobacilli would be a bunch of rod-shaped organisms joined together in a chain. Round organisms, or cocci, can make what we call a staphylo or a cluster arrangement. So staphylo is also called cluster. This is not found in the bacilli. It's just found in the staphylo, or I'm sorry, in the cocci, and it makes what we call a staphylococci arrangement. In the bacilli, we can also find what we call palisades, which is kind of like a picket fence type arrangement. So this is only found in the bacilli. Now, just like with shapes, there are other arrangements as well. So there's what we call sarcinae, which is like a cube. There's tetrad, which is a square, essentially. In the bacilli, we might also find what we call the V shape. So I'm just going to draw that here for you, where there are two bacteria, but they're not joined end on end exactly. It's where they kind of attach to each other a little bit at the end, but not in like a linear fashion, more in this shape where it makes a V, like it says. So this is called V shape, and that's only found in bacilli. However, again, just like with the shapes, the most common shapes that you're going to find, especially in the healthcare professions, are cocci, bacilli, spirillum, and spirochetes, even though there are others. Kind of the most common shapes that you're going to find are the diplococci, the staphylococci, the streptococci, and the single bacillus, diplobacillus, streptobacillus, palisades, and sometimes V-shape. So, like I said, there are other shapes, there are other arrangements, but our most common shapes are the coccus, the bacillus, and the spirillum and spirochetes, and the most common arrangements are going to be the diplo, strepto, staphylo, palisades, and sometimes V-shape.